Hello students, welcome to Skill Area 1 of the Basic for Microbiology module. Food microbiology is the study of microorganisms in food. It is very important to know them because it can help us understand their use in food preservation, food fermentation, understand food spoilage and foodborne illness. There are three main types of microorganisms that you need to know, and it's very important for you to know how to draw and label them as well. There are bacteria, fungi, and virus. Drawing these structures can be quite challenging. However, you can do a lot of practice in order to be familiar with them. Make sure that you can draw and label them, and most importantly, understand the function of each of these parts. The first part we have is the capsule, which is the outer protective layer of the cell, followed by the cell wall, which is the inner protective layer of the bacterial cell, and then the plasma membrane, which controls the transport of material in and out of the cell. The small dotted points are ribosome, which is the power of the bacteria, and the white region is the cytoplasm, where all the metabolic reactions occur. DNA, which contains the genetic information of the bacteria. The tail-like structure is the flagellum, which is to allow the bacteria to move around. And the small hair-like structure is what we call pili, which is to attach the bacteria to the host cell. Drawing the structure of fungi is quite simple because it is a very simple microorganism. For viruses, you need to know how to draw them properly and understand their function as well. The outer pointy thing is what we call the envelope protein, which is to help the virus attach to the host cell. The inner layer is what we call the envelope, which helps to mimic the host cell membrane. And the red part is the viral tegumen, which contain protein that help in RNA replication. The shell-like structure is what we call capsid, which is a protein called to protect the viral genome. And the inside is what we call the viral genome, which contains the RNA. Classification of bacteria can be divided into four different features. Particularly, the first one is the gram stain test, in which you can divide bacteria into gram positive, which appears purple or blue under the gram stain test, or gram negative, which appears red or pink. It is very crucial for students to be able to draw the structure of the cell wall of the gram positive bacteria as well as the gram negative bacteria. The first layer that I have drawn here is actually what we call the cytoplasmic membrane, and the purple layer is what we call the peptidoglycan layer. If you see the difference between these two, gram positive have more peptidoglycan layer than gram negative, and gram negative have two more extra features, which is the lipopolysaccharide and the outer membrane, and they only have a single layer of peptidoglycan in their structure. The bacteria can also be classified based on their pH growth. You must learn to try to draw and understand the graph of this pH growth rate. There are three: acidophile, neutrophile, and alkalophile. The third one is the temperature growth. Again, students are required to be able to draw this graph for their examination. We have saccharophile, mesophile, thermophile, and hyperthermophile. If you see the peak of each graph, it corresponds to the maximum growth rate, where the temperature is what we call the optimum temperature. The final classification of bacteria is based on their oxygen requirement, where they are mainly divided into 5 different types of oxygen requirement, which is obligate aerobes, obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and microaerophiles. Each group has their own criteria for them to grow. Remember, this sign is indication that it's a biohazard sign. Please understand the proper PPE to wear in the lab. It's basically safety gloves, safety goggles, lab coat, and closed shoes. That is the end of the scale area 1.0. Good luck for your revision.